diffusion model for single cell drug response prediction. So first, let's start by reading the abstract. Has everyone read it? So what do you guys think is the gap sentence in this abstract? There is actually no sentence, but they just introduced what they are going to do in the first sentence. And what do you guys think about the abstract?
Easy or typical? Very clear or not clear? Who do you think are clear? Hands up? Or confusing? Hands up? Yeah. Good. What do you think?
Yeah, it was actually really hard to understand it. It took me some time to actually get what they were doing. And if you don't know any guidance here, you don't really understand what they are doing. And if you're a biologist specifically, you can't really understand.
So indeed, one reason is jargon. Another reason is low gap sentence. Well, let's think about their first sentence. They developed the single cell perturbation prediction diffusion model, the first diffusion-based framework. That means there is some other framework for this problem.
Yes.
But diffusion model is first time. That's the meaning of this. Clear? Then what kind of 뭐가 막 궁금해지나? What kind of information then is missing when you see this? Okay, this is the first diffusion-based framework. What's your question come in your brain, in your mind? Why? Yes. Why do we need a diffusion model? Listen. Kind of two things come in your mind. One is, what kind of previous approach have been used? Maybe XGBoost or simple linear regression or whatever. And then second information we need, what is the limit of the previous approach? If previous approach works very well, then do we need a diffusion model? No. Here, why this abstract is not good is we cannot distinguish these two things. And so now the diffusion model is very popular. That's why we use it. Or previous approach doesn't work well, so that's why we use the dispensing method. These two are completely different. Which one is better? Yes. That kind of. But if you don't write this gap sentence, then people can think that, new intern comes. So, okay, nowadays diffusion model is popular. Why don't you try it? this is current abstract. That's why this is not a good abstract. Okay, so no gap statistics. That's why very risky abstract. Where they publish it?
It's actually they submitted it to IEC LR conference.
Oh, conference.
So it then
Okay. Good. Okay. Why don't you move?
So basically, the first question that comes to mind is that why is drug response prediction hard? So basically, when we see a cell has a lot of genes, like it can have more than 200,000 genes. And if drug or dose is applied to it, then there are a lot of possible combination of changes that can occur. Like it not only affects one gene in the cell, it can affect the thousands of genes in the cell after the treatment. So, as a result, what happens is that the genes don't change separately. They can change coherently, and which can cause difficulty in checking all the changes that are occurring in all the genes. And the other reason is that there are many cells and many drugs and also many doses. So there are a lot of possible combinations that can form, but there are only a few observed or experimentally proved fraction of these combinations. So we have a lot of unseen combinations and unseen drugs. So another reason is that when we apply a dose to a cell, it should just affect it magnitudely, like it should just change the effect of the dose, not change the direction of the effect. Like it should, if it has to move in the positive direction, then it should move in the positive direction. And also, if we look at the previous models, they use the baseline, that is the that is a cell that is without any treatment, then drug and dose as the input. But these inputs are entangled, like they put it as input, but they don't know how each of these elements is affecting the output that they get. So the reason was that for the how the existing models are not working well is that they do not basically disentangle the effect of these inputs. So given these challenges, we need a model that can generalize beyond observed data. It should also work on the unseen data. It should also separate the baseline state from the drug-induced cell state. And it should also have dose-aware control, that how much dose is affecting the cell. And it should also preserve the cell identity while applying the perturbation. It should not change the cell's identity. So in order to overcome these challenges, the authors introduced the single cell perturbation prediction diffusion model. So first, I will introduce you the basic components of the model. Basically, there are three coupled components. First one is the latent space diffusion backbone. That is basically the backbone of this whole architecture. And then we have the condition representation and GD attention injection that is going to be applied for the denoising step. And then we have the four-state training and factorized guidance.
So you should explain. Okay. Slow.
Okay.
No one can understand this whole.
Basically, I will actually explain these one by one later.
Okay.
So this is just the overview of the structure, how it goes. So basically, the single cell PPDM consists of three coupled components. First one is basically the latent space diffusion backbone. If I tell you simply in this step, they just are going to use the gene and add noise to it and then predict the gene set by denoising it simply. And the second one is the condition representation and GD attention injection. That is basically going to be in this condition information that will in turn result in the denoising of the network. And then we have the four-state training and factorized guidance, which is basically going to be here. This is the condition that is going to be injected into the GDA. So basically, they are going to train the model using this information. So for the first step, we have the latent space diffusion backbone that learns for post-perturbation dynamics in a shared VA space. So in this model, authors think of the drug response should be treated as a causal intervention acting on the baseline transcriptome. So here we have X pre and X post, that is basically the pre and post perturbation expression over G genes. And then we have D, that is basically drug, and it contains the information of the structure of the drug in the form of SMILES, that is basically the string. And then they also have dose information.
What do you mean that? What is D?
D is basically drug, and it contains the SMILES, that is basically the chemical structure in the form of the string of the drug, and then the dose information of the drug.
Oh, can you tell what is SMILES? Okay.
SMILES is basically if we have a chemical structure. I actually described it in the previous general club too. So basically, it is if a drug every drug has a chemical structure. So in order to present it easily, it is written in the form of a string, and that is SMILES, in order to use it in the model easily.
So SMILE is basically how to write down the chemical structure in the text.
Yes. So then we have T dot. It is basically it is representing the causal interventions of the drug. So this is the most important part of the this equation. So basically, if we look at the architecture of the backbone now, we can see that we have first step is like we have gene expression. It contains whole gene expressions of gene expressions, and again, it is actually after perturbation that is exposed, which is this one. So they apply a pre-trained similarity VAE encoder E. It is actually already available framework. They apply it and convert it into the latent vector, that is C0. So at this stage, the whole gene expression is converted to a more concise latent vector. So after that, once we have a clean latent vector, at that step, the noising is introduced. So basically, here we have the diffusion process, and they introduce the Gaussian distribution noise into the framework on the latent vector. So here, if we So basically, here beta T is how much noise is added into at each step, and alpha T is basically 1 minus beta T, which is the fraction of signal that is preserved or at each step t after adding the noise, and alpha t bar is actually basically the product of all the previous steps of alpha t. So after this, we have a denoising process, which is basically simple if we think of it as it is just a neural network. So in this neural network, they add the noisy latent vector with the time step embeddings, and they get the predicted noise TT. So actually, we will go into detail about this conditioning information on the later, how this is introduced into the denoising step. And then we have the gradual denoising of the noisy vector. And then after that,
So Aqsa, go back to it again from the beginning, step by step. Don't do this. Really explain step by step. Don't read the alphabet. Try to explain the meaning. So you plug in the data and then
yes, so the data at this step which is treated with the drug already.
cosmic.
Yeah.
Cosmic.
Oh, okay.
Where are you explaining this?
So here we have, first step is the gene data that is already treated with the drug. So basically it is for the, and it is called ex post, as already I explained here. So after that, this data is added to the encoder that is pre-trained similarity VA encoder, and this is already existing technology, so they only used it. So in this step, the encoder acts on this gene expression and convert it to a latent vector that is easier to handle. That is D0. And after that, the diffusion process starts adding noise to this latent vector. So in this equation, basically it is the Gaussian distribution noise is added. And basically what is happening here is beta t is noise schedule that is not added here, but alpha t is basically 1 minus beta t. That is actually the fraction of signals that is preserved at each step t. And alpha t bar is actually the product of all the previous steps that are being added into with the noise added.
Alpha t is between 0 and 1. So long as it multiplies, the signal it goes to 0. And 1 minus alpha t is keep increasing. That is multiplied through the Gaussian noise, so that the noise keep increasing.
Alpha t bar is keep decreasing to the 0. So that means that Gaussian becomes n 0 comma I.
Yeah.
And become pure
noise. Yeah.
So when we reach there, we reach to the ZT, RZT, right?
Yes.
So when we reach to the ZT, it becomes nearly pure
noise.
Then
so after that, then denoising process happens, in which we have epsilon t, that is basically the predicted noise. And here we have with B theta, that is basically the just a neural network. And we enter the noisy latent vector into it with all the time embeddings that happened previously. It is in the form of sinusoidal information.
What's gamma T?
So gamma T is the embeddings, time embedding.
Okay.
So basically, when they added the noise, they also saved the information of the time at which the noise was added. So they used that information and added it this time.
2000.
Yes.
So even the time step embedding is it's confusing to me. So if we know the ZT and gamma T is ZT minus 1, GT minus 2, GT minus 3, that's what it is.
So basically, it is just taking in account how much noise has been added, not knowing the exact how the structure, just keeping in track how much the noise was introduced.
The how much?
How much is more like, it is like, it is basically, you can think of it as T. That is the total.
Anyone who knows what is the meaning of time step embedding?
It's sinusoidal.
Yeah.
Yeah.
Why would they use sinusoidal?
The sinusoid they usually they use to tell the time information. Just so So, what this gamma t means, the diffusion process is 0, 1, 2, 3, 4, 5. Let's say large T is 1000. Okay. Then if we tell that to the neural network that between 1 to 1000, if we know that from the beginning to the end is 1 to the 1100 step, then we know that it's early stage. And 900, we know that it's late stage. We know the 1000, but they don't know. We need to but if, for instance, if this step is a 10,000 step, then 900 is later stage or earlier stage? Earlier stage. If it's a 1000, then 900 is later stage. So the number, actual number cannot tell that we are in the early stage or late stage, right? So we need some common way to the neural network that we are in the early phase or later phase. One way is using the sinusoidal timer. So sine, if we're mixing the sine and cosine, everything is between 0 and basically 2 pi 9 points. So then using the sinusoidal way, we can tell that we can define the beginning and ending information. That's typically this AI field using the sinusoidal thing to tell where we are. We can use this kind of idea for any data set. For instance, your sleep time. You begin sleep and then you sleep like 8 hours. And we know that 7 hours is quite late stage. How we can tell this to the AI? It's again expressing from the beginning to the end of sleep as a sinusoidal information. So that's why I think this you can just think that this gamma t is some where we are between this from the beginning to the end. So that if here is only, that means noise is pretty weak, and then later we can tell that the noise is pretty heavy. That's the why they call the embedded timestamp. Okay, move forward.
So they perform the gradual denoising using the time step T, because it is like an iterative process to remove the noise, and then we get the VVO tilde, that is basically the generated latent network that is clean of any noise. And after that, this clean generated latent vector is added to the pre-trained similarity via a decoder, and it decodes it back to the generated gene set. So basically, in this backbone, here the model is basically just learning what treated cell looks like in general, and it doesn't have any information yet why they look like that way. So by learning the denoise-noisy latent representation, like without any conditioning, it is basically just providing a foundation onto which baseline information and the drug information later can be injected in and controlled. So the next step is basically injecting these doing these conditioning and injecting it back into the denoising framework. So for this one, we have basically two types of encoding. First one is the state encoding, and which is basically just the information of the genes without any treatment. And the encoder is similar to what previously used. And here we have XPRE, that is the cells without any treatment. So what this ZPRE is doing is that they have, so using the encoder, they changed XPRE, that is basically the genes before any treatment, into the latent vector, similar to previously what they did with the drug-induced ones. So they are using the same encoders and same decoder here. Next, they apply the conditions on the latent vector using the posterior mean. And here we have the expected value of it, and it is basically the Gaussian distribution. And then the second part is drug encoding. So in drug encoding, they have a drug, and they use the SMILES string of the drug through RD-KIT and convert it to the Morgan fingerprints. That is basically just converting it to zero and ones, simply for easier usage. And they then add this to the MLP, which is multi-layer perceptron. That is the simplest form of drug representation. And then this drug is added to the film, which is basically the feature-wise linear modulation, and it just scales it and doesn't change the direction of the Z drug. So then we have the condition injection, which is basically just getting the conditions from both drug encoding and state encoding and combining it and injecting it into the GD attention. So when they get the condition and they inject it into the GD attention, first step is the self-attention that basically just refines the latent representation. And then we have the cross-attention that couples the latent ZT with condition token that it is getting from here. And then it has the fill plus residuals and their norms, and then it converts it back to ZT, that is without ZT minus 1, that is denoised version of it. So for this condition, it will be explained later in the training step more in detail. So basically in this step, they just decided where the conditioning is entering and how, but they didn't give much condition at this step, and they didn't tell really that how much this condition matters. So this injects basically only the baseline and drug information, and that we still don't know the disentangle or the control. We can't really control the effect of drug or base till now. So this will be addressed in the training and guidance strategy that is in the component 3. So for the training, basically it is a
Yeah. Slide.
Yeah.
Yeah, please ask the question. If you're confused. Everyone? Any questions?
So could you explain the meaning of second box, the second blue box?
this the purple one.
The condition.
So basically they are just applying the conditions. So conditions are not yet defined here. So there are 4 different types of conditions that will be explained later. Yeah, so they are just using the mean of the posterior mean. Yeah, posterior mean of the encoder's prediction that it does. But here it is like,
So what is the purpose of that?
so basically the purpose of that is to make the encoder learn about each condition.
First.
Yeah, I think so. Because like there are 4 conditions.
So you find that function is
The posterior,
differ from it's the same or
it is from the encoder.
from the so can you say that the previous diffusion model and that this module shares the same?
So it is the same backbone, like the same encoder that it use.
Yes.
Like they use already pre-trained similarity, so
Okay.
that is same.
That is same.
But the information is different here.
Formation.
Like that encoder and decoder is now gone. Kind of like they add new one that adds the condition. But it is the architecture is different. Yeah.
Same architecture but different
Purpose.
purpose. So it trains separate.
Yes.
Then is that so you write you find there that represent that.
Yeah.
Function. So they mix the information.
So basically it is, I didn't really get a lot of details about it. They just mentioned that it's the QN function that just combines the information that is coming from the drug encoding and like the state encoding. And it in some cases, like if we look later, there are 4 conditions. We can have both information from both drug encoding and state encoding. And then we can have only information from one of these. And we can also have information from none of these. These are the 4 conditions basically. So that is what is going to into the F.
Maybe now can overview the over.
So
Step by step. More time.
So basically this step starts after the noise is introduced into the clean light interface that we have already seen, that is ZT. So for this purpose, they add 2 informations. That is basically one is coming from the state encoding and the other one is drug encoding. So state encoding is basically the set of genes that are not treated with any kind of treatment. And it is similarly converted to the latent space as before. And then we have the drug encoding. That is basically the information of the drug that is converted into zeros and ones from using the MLP. here based on. No, so the fingerprinting is used to convert it from smiles to the 0 and 1s, and then MLP is applied to get the continuous drug representation. And then this information from both of these encodings is added into the fusion function that is in turn make the condition. And this condition is then inserted into the GT attention. So this attention architecture has 3 steps. One is self attention, and then we have the cross attention that basically just couples the ZT with the continuous.
For example,
So I actually didn't really get into the detail. It is the simple attention framework. So it just defines the latent representation. It is actually really detailed. Very young to study this.
okay, very important.
Then they have the cross attention. So in this, they have information from the latent CT and also this condition C. And then they have the pill that has the information of the drug and also dose. And then they have different layers. And this is how the diffusion just goes on till it is denoised.
Anyone who knows what is a Q and K and B in this word? In this slide? Anyone who knows what is Q and K and B?
Already there. Is A is a key and place of charity. So we want to find the value from the you want to update the value.
원래 오리지널 셀프 로테이션 모델 같은 경우에는 인풋 데이터에서 이제 퀄리티 밸류를 찾으려고 하는데 여기서의 그 인풋 데이터에서 쿼리만 그다음에 저기서의 포지셔널 인젝션의 인포메이션, 스매틱한 벡터를 가지고서 퀄리티 밸류에 해줌으로써 뭔가의 그런 스매틱한 정보들을 이제 멀지를 시켜주려고 하는 모델인 것 같습니다.
그냥 비노이징을 하는 게 아니라. 뭐, 힘들는데 이거 뭘 그렇게 하는 거야? 어렵지 쉽게 설명하는. 나중에 넘어봅시다. 그러면.
So now we are going to move towards the training step, that is 4-stage training, and it basically is teaching the model to separate between the baseline and drug effects. So there are 4 conditions. One is in which we have both baseline cell and all information and also the drug information. Second is that we don't have cell, we just have drug. And the third one is that we have the cell, but we don't have drug, and there is a null case too. So in order to be able to make the model predict even when there is a missing data. So basically, if we look at the figure, this is the step where the conditions are being applied. It's purple box. So this is, if we look at the top one, it is just with the baseline information. And then we have this one with the both. And the last one is just with the drug information. So this is how the condition is going into the model. So they basically calculate the loss term for this, in which they use the loss of denoise and the loss of inject. So what is the loss of denoise? It is basically just using the information from the noise and also the conditions and just injecting it back into this total loss. And then we have the inject loss, that is basically combining the aligning loss and the information loss. So I will explain the information that it is getting from the aligning loss and the information loss. And aligning loss is basically, it has the, so it has the information for each sample that is normalized to the empirical response, that is basically this. and it gives the direction of the sum. And then we have the W matrix, and this whole information is aligned with it. So basically they align the whole matrix with this information to keep the track of the negative losses. And for the info loss, the basic idea is to preserve the information of the cells across. No. So it is basically to provide the information to separate the drug. Because yeah, it has the drug information and cell information, and this T is temperature. So the align information basically gives the direction, and then the loss information gives the information about the drug effect. So when these are combined, we get the loss that is injected through the GD attention, and when it is combined with the denoise loss, this is the total loss that we are getting at the end of the 4-state training.
Sorry, I missed the. So here, this is a training process,
Yes.
and do we train the same model or the process?
So I think the same model, but with 4 different conditions.
For different conditions?
Yes.
They just put only pre or just drug information, then both step by step, or just
So it is trained on all 4 conditions.
conditions?
So basically later there is an equation that shows how each condition is affecting the inference. Later.
For this kind of training, they added another module here. But what is the feedback?
So here, this part, the one that gets the condition into the model. The model is the same, but based on the condition, the results are
Is it inside of the
denoise. Yeah, that one.
the previous?
Yes.
So this is the kind of details of that
The
feedback.
conditioning, yeah. Yeah.
Explain. Sorry, one more time, but what is
So it's okay.
here?
It's okay. I also had really hard time getting it to know this paper. So what should I explain.
Yeah, so, uh,
Before that, could you explain what is the purpose of L align and L
right.
info?
So for me, only the LED noise is enough for our aim, but why then else is it?
So it goes back to the first slide where we had like we need to have the information of the drug and the state and how it is actually affecting the output.
Right.
So that is why basically the loss of info is based on the drug. And align is basically, I think, the direction of the drug, and that one is
Direction of draw.
it's
Direction of the drug means you wanna like preserve the qualitative effect of drug.
the drug, yeah.
Using syntaxes. Okay. They construct key and value. What is the
For also, yeah.
Is W using other parts or only for
Like for this part.
only for this?
Yeah.
모델을 구하기 위해서 새로운 물건과 어떻게 모델을 만들고 뭐 하자. 지금 W가 장래 안 쓰면 W를 할 이유가 있네. 굳이 또 학습을 해서 이렇게 되는 거죠. So my point was to make this,
Yeah.
they added another kind of module or function W here.
Yeah.
And what's the point of
They mention it like it is kind of a learnable metrics, but I was also not really sure about what they meant by that.
maybe the W size is quite small.
Oh.
Then maybe they want the T draw has enough information to identify the direction.
Hmm.
Yeah, maybe they give another restriction. I'm not sure. In that case, I think they need some restriction for W.
Hmm.
And if they give, this should give some information.
they use first. They produce and
Yes.
what is the they they same information for every stories that they see vector to convert the CDT to CT minus. They use that information to every step. T minus 1, 2, T minus 2.
According to what we mentioned in the predictives, yeah.
Customers, they use the same information, same C vector.
Mhm. There will be a difference between the previous.
C vector.
Ah. Yeah, the C vector.
See,
Hmm. But I think they just add it once and then denoise it later.
either put in the slide or
Because
use this structure for every
Wait.
stories.
like for
T minus 1. Yeah.
this one. Yeah. What I thought was that maybe they are just adding condition once, but they are because the data is changed at every denoising step. So that is the only thing that is changing. Conditions are the same. It's just my opinion. I think the W is related to the GT attention maybe because they are like injecting it into the attention mechanism. So W has to do with that.
W is from, I think W is from stable diffusion. I can see there's a reference paper that's called high resolution image synthesis with latent diffusion. That's the original paper of the stable diffusion. I think the entire structure referenced in that paper. Can you please find the Google the stable diffusion original paper, please?
Should I Google it here?
Let's Google it.
Is it okay?
The point. It is originally for the same diffusion, and most of the clients study on this diffusion model based on the mean diffusion. Most all of the generating models and for text and images generation are based on these models, and anything that condition models that we are very confused on is based on this part, and they applicate they modify and applicate to the RNA structure. So they during the process of the modification, it becomes very vague and difficult to understand. So I think if you study more about this conditioning part, you can explain the whole structure very easily. So yes, I think we need more study on diffusion to explain the applications of it. That's why I think. In the computing part of the document is explained on the this part. I guess if they use the similar index, W part is explaining this WQK. W here, yes. Also the learner These are learnable process projection matrices. For visual detection, they say visual detection because this paper is based on image generation, but in our paper, this can be interpreted as differently, but I'm not sure about that actually because I am from my knowledge. So it's so we have three interpretations. Yeah, in the those in the paper they use here. Same structure, they use QKB, also the attention model. So they use W as a large matrix inside of the latent space. So between the diffusion process, between two diffusion process, they use attention model to input our knowledge, the condition model matrices, to learn more about the conditions inside of the latent model. So they use this structure, and this structure is kind of standard for diffusion models. This is just like intuition. Perfect. And first, so here, this latent vector, let's say that it contains the information about this post perturbation, perturbed gene expression level things. But it adds noises and until it goes to some random noises. So maybe we can interpret this process as we just lose that data or latent state loses some information. So step by step, you lose some information, become noise, lose some information, become noise, and more like noises. And denoises is adding some, recovering some information. Then to recover it, we need some information,
And what we are going to do is we will get the information from the that gene expression data and also drug input. So this, and so in summary, this model is asking this: can the previous gene expression level information and drug information recover the losing information of the post data, and step by step.
So one more thing is that why we dissect this way is just think about it. So I want him to serve the differential equation problem. I want him to know how to solve the differential equation problem. But can I make him do that immediately when he was a freshman? No. We have to give him easy problem first, and then slightly more, and we can reach it. Then let's think about it. This is completely noisy state, GT. And G0 is our original state. If we give a AI that from the completely pure noise to the original answer, is it easy problem or difficult problem? Very difficult. So even AI, it's too difficult. But if we dissect this as a 1,000 step, completely noise and a little bit signal and noise, then this is much easier problem. So the key point of this diffusion process is, but how to make that means we want to make it from the noise to the here, and then we want to dissect the problem to the step by step. There is small problem, and then small problem, small problem, small problem, small problem. But previously, and how to make this is small, how to dissect is very, very difficult. So I wanted to teach the differential equation, then from the base to here, how? First, what we have to teach, maybe X, Y, Z, and then first order differential equation, and then maybe second order. There is some rule, but for the data, how to define the easy to the how to dissect it, it's very difficult. But diffusion model was a very smart way. So that's why diffusion model become very, very popular, because this is a very common and simple way to dissect the learning the something from easy. So rather than learn this pattern, we will ask him learning this. If we know the easy part, then he basically know everything. Doesn't make sense. So that's the key point of diffusion model. And you may go ahead.
And so that means we are going to use the G information and drug information to learn some function that recover that information. But maybe the simplest way is just concatenate that G information and drug information and just plug it to some kind of neural, like the neural network. But it has some, I think it's too naive, and maybe they want to see some interaction effect between these two variables. So, for example, because we don't have full data, so for some we only have some drug information, and certain drug we got G post that A expression level is very high. But actually, the drug can decrease expression level because the XP was too high for every data. So maybe it learns some.
Can you tell me one more time that?
Okay.
Or you can write down whatever. Yeah. That's the key point, I think.
And free data. Okay. So we have drug
one information, and for there are several posts. One post two, and gene maybe gene A is very high, maybe 100. Gene A 100. But actually, this was due to in pre-data, gene A was 1,000. And maybe depending on this, the 500, and so this could be 300. And depending on this, the context, that drug they should learn that, oh, okay, depending on the state, drug one can increase or decrease gene A level, but due to this information, so it may learn some shortcut. If gene A is 100, okay, it uses third one. What are the post 1, 2, 3? It's different. Maybe it's drug one is applied to cell type 1. Cell type 1, cell type 2. So my point is, when they want to cover the information, they would just want to, this model could learn this very easy correlation, but they should consider some context of this pre-data. Depending on pre-data, the drug one can have different effect. And in that case, maybe so unseen data is coming, then maybe because this model learns very wrong, just correlation in the present data, it will generate some wrong prediction. So this is why we need to force this model to consider both this and drawing. To do that, I think they use the attention. So maybe if I had more, I could generate more good example for this. 그냥 뭐 생각해볼까? 그러니까 이걸 왜 하는가? 생각해볼까? 얘는 약을 줬을 때고 풀이면 그 전 상태가 있다는 거거든요. 그럼 그거의 차이가 약이 똑같으니까. 그죠? 엑스프리가 있고 엑스포스트가 있으면 내 빼면 그 약 넣었을 때 효과죠. 우리 약 효과를 다 아네? 그러니까 주어진 약에 대해서 다 아는 거잖아요. 100종류 약이 있으면 각각 100종의 약이 그런 걸 다 아네? 그럼 우리 이거 왜 하는 걸까? 우리 이미 다 아는데 몇 개. 이걸 하는 이유는 101번째 새로운 약이 왔을 때 어떤 베이스 정보를 줬을 때 걔가 어떻게 될지를 알기를 원하는 거. 그걸 하려면 지금 이 짓을 해야 된다는 거. 자, 그러면 다시 100개로 돌아가 보자. 그럼 100개 각 약 1번에 대해서 두 벡터의 디퍼런스가 있겠죠. 그러니까 모든 약에 대해서 그 유전자 뭐 1000종류가 있으면 각 1000종류가 어떻게 바뀌는지 다 알긴 알아요. 그걸 알면 101번째 약이 왔을 때 그 정보만 가지고 예측할 수 있을까? 아나 모르나? 황규영이라는 약을 쓰면 어떻게 세포가 바뀌는지 알아요. 여기가 알아요, 여기가 알아요. 네 번째 이 친구를 쓰면 어떻게 바뀔지 예상할 수 있는. 사실 그냥 뭘 알아야 할까? 어떤 거를 우리가 알아야지 네 번째 약이 왔을 때, 아니면 이렇게 해볼까? 좀 더 쉽게, 그냥 이렇게 해봅시다. 황 박사님의 국영수 성격은 나는 알고 있고, 이 친구 국영수 성격을 알고 있어요. 이 친구도 알고 있어요. 그러니까 황병 박사님이라는 선생님을 붙였을 때 어떤 학생의 국영수 점수가 어떻게 바뀔지 우리는 알고 있고, 여기도 알고 있고, 여기도 알고 있어요. 네 번째 선생님이 등장했어요. 이 친구를 투입하면 이 학생의 성적이 어떻게 바뀔지 예측할 수 있는가. 그 문제를 지금 풀고 있는 거 있지 않을까요? 방법이 뭘까?
선생님,
음,
1, 2, 3번 선생님 좋아하는 거.
그렇지. 가장 중요한 질문 그거지. 관상을 일단 딱 보고 있잖아. 혹은 뭐 관상이라고 표현했지만 이 친구가 거리로 뭐그 히스토리가 있을까? 왜 관상이라고 표현하면 여기는 이 친구의 화학물의 구조를 쓸 거거든. 스마일스라는 게 화학물의 구조니까. 그러면 이 친구가 설명을 딱 봤더니 그나마 얘랑 제일 닮았구나. 그럼 얘랑 비슷할 거구나. 혹은 대충 보니까 여기에 조합이 얘 절반, 황 박사님이 0.25, 여기 0.25 섞으면 대충 이 관상이 나오네. 그럼 어떻게 예상하는 게 가장 확률적입니까? 0.25, 0.5, 0.25 섞으면 되는 거잖아요. 여기서 아까 QKB라는 게 나왔는데 그 KB에서 K랑 B가 하는 역할이 뭐냐면 K는 그거예요. 예를 들어 황 박사님, 그리고 두 번째 학생, 세 번째 학생. B는 뭐냐면 황 박사님을 쓰면 국어 영어 수학 점수가 얼마만큼 바뀔 건지. 그럼 이 네 번째 학생이 왔을 때 K 밸류가 있을 텐데 그거랑 이 세 명 사이의 관계를 보고 이 세 명의 부위를 잘 조합을 하는 거야. 그러면 이제 우리는 뭐만 알면 이제 그러면 이 친구를 이제 예측할 수 있을 거냐면 음, 가장 중요한 게 그거지. 두 가지가 필요하지. 황 박사님을 집어넣으면 국어, 영어, 수학 성적의 학생이 어떻게 바뀔 건지 그 B 값을 아는 것 꼭 필요하죠. 그게 틀리면은 말짱 꽝이니까. 두 번째는 K. K 값은 뭐냐면 이 사람 세 명의 특징을 관상. 그죠? 그리고 이 사람이 이 세 명이랑 관상이 얼마나 달라있는지 이게 K 값이다. 근데 그 K랑 V를 우리는 뽕으로 얻을 수 있나? 쉽지가 않아. 그냥 구조가 비슷하다는데 뭐 어떻게 저 이걸 어떻게 근데 사람마다 다 다르겠지. 뭐. 알파벳이 비슷하면 약간 다 비슷하지. 근데 우리 목적이 있거든. 어떤 목적? 약의 효과를 어떻게든 잘 예측하는 거. 즉 이거. 이 문제를 제일 잘 풀 수 있도록 하는 K랑 V를 찾는 게 목적입니다. 그래서 알아, 우리 원래 약은 100개 있잖아요. 근데 100개 주어진 것 중에 이렇게 90개만 가지고 하겠지. 그 다음에 이제 10개를 가지고 이 방식을 썼을 때 과연 잘 되면 이제 그 KV가 우리는 원하는 K랑 V인 것입니다. 일단, 음, 음, 음. W가 어디니? 가볼까? 내가 여기까지 생각하느라고 맨 마지막 거 들어볼까?
여기 W 말하는
요거?
이 W는 좀 다른 애일 것 같은데 얘는 어떻게 보면 약간 레이턴트 드럭이 그러니까 그냥 스포츠가 방향이잖아. 방향 정보를 좀 가지고 있도록 강제하는 역할을 하는 거야.
좀 내가
텐션하고는 좀 반대가
없는 거. 근데 논문에서는 그렇게 하긴 했었거든. 에러 라인은 뭐냐면 비슷한 놈은 비슷하게 만들고 때림표는 약간 반대처럼
얘랑 다른 애들은 다르게 해서 그러니까 어떻게 보면 스페시피시티가 되게 높아지도록 만드는 건데 결국에는 이 방향을 얼마나 잘 얘가 정보를 갖고 있게 만드냐에
W가 관상? 아니지. 관상이 비슷한 애들은 비슷해. 아니면 성적이 비슷하게 만드는 선생님들은 비슷하게 아닌 건 아니게 만들고 관상이야 아니면 결과일까? 그러니까 이거를 온라인 시킨다는 게 비슷한 애를 비슷하게 만드는 방향인 거야? 아니면 성적이 이제 국어 스페셜라이즈, 수학 스페셜라이즈, 과학 스페셜라이즈 그걸 온라인 시키겠다는 거. 항상 그 두 개만 생각하면 될 거야. 그러니까 X랑 Y잖아요. X는 관상이고 Y는 이 선생님 투입했을 때 점수 잘하는 그 Y인데 X를 온라인 시키고
이게 지금 거기서 한 가지 더 부담을 주는데 지금 두 가지 종류가 있는 거예요. 그러니까 사람 개인 뭐 관상이 있고 약간 성적표 종류가 따로 있어요. 그걸 어떻게 잘 선생님의 성적 과거 성적
그러니까 성적 어떻게 올렸는지 Y.
그죠.
선생님
그게 Y 말고 그러니까 X하고 X2가 있는 X1이랑 X2가 있는 그러니까 두 개의 정보가 있는
뭔데
그러니까 여기서는 이제 진 정보랑 그러니까 포스트리에 진 정보랑 양분 정보가
그치
두 개를 잘 조합을 해서 만들어야 되는 상황인데 뭐 관상하고 예를 들어서 뭐 선생님의 이제 그러니까 Y가 아니라 다른 어떤 정보가 더 있어야 됩니다.
여기서 말하는 나는 엑스프리 포스트는 나한테는 어떻게 보면 다 수학과학 점수들이고
점수
그리고 약은
아
그 선생님의 관상인 거죠. 왜냐면 구조 정보가 있습니다.
그럼 관상이 맞고 관상이 맞고 프리는 그러니까 어떻게 보면 오르기 전에 성적 이미 잘하는 애들을 올렸으면 사실 의미가 없는 거잖아. 그런 애들을 좀 구분을 좀 지으려고 이런 거 관상이랑 그런 거 좀 구분을 좀.
그러니까 이렇게 이해해볼까? 예를 들어서 너희가 수학을 엄청 잘 가르쳐. 얘도 수학을 엄청 잘 가르쳐. 세 번째, 네 번째 다 수학을 잘 가르쳐. 김현 박사님까지. 근데 이 5명 우리가 보잖아. 비슷하게 생겼어? 안 비슷하게 생겼어? 미안하지만 하나도 안 비슷하게 생겼어. 우리의 관점은. 근데 우린 AI한테 뭘 가르쳐줘야 되냐면 이 5명은 똑같은 애다. 똑같은 애다. 똑같은 애다. 우리 목적은 그냥 수학 점수를 올리는 선생은 다 이렇게 생겼다라고 알려줘. 대충 이 5명에 대한 그 정의를 해놓고 그래서 여기가 왔을 때 이 친구가 이 5명이랑 그 관점에서 비슷한지를 판단해야 돼. 아마 그걸 하겠다는 거 아닐까? 그러니까 그 K를 러닝한다는 게
네.
그러니까, 이걸 잡으면 우리의 목적은 그 약효 예측이고 예측을 잘하기 위해서 약들 사이에 비슷함을 잘 정리하는 거고 그걸 하고 있는 것 같다는 생각이 드네요. 일단 내 느낌은.
아마 이 지는 그 로테이션 들어가기 전에
그래?
네. 지는 아마 걔는 아마 여기서 이 다음에서 뭐 어떻게 하는 것 같은데
아니면 진짜 약의 반상인가 이거는?
그렇게 봐야 돼. 진짜 약의 진짜 약의 반상 그러니까 이런 느낌이니까
근데 여기 또 약의 효과가 있잖아.
네. 그러니까 반상이 뭐 예를 들어서 안 좋은 사람도 성적이 그러니까 결과값이 되게 높아요. 근데 이 사람은 이미 잘하는 사람한테 들어가지고 그걸 사실 별로 능력이 없는데 잘하는 것처럼 얘가 생각할 수도 있잖아요. 그걸 방지하기 위해서 이런 제약을 택 걸어줬다라고 저는 이해를 좀 했어요.
아니면 이걸까? 약간 이렇게 하면 이해될까요? 이것도 내가 말한 그걸 하는 것 같아. 그러니까 진짜 데이터는 100세트밖에 없으면 그 100세트를 가지고 일단 관상을 그죠? 온라인을 시켜주고 근데 사실 100세트만 가지고 그걸 다 러닝하는 건 사실 불가능한 거잖아요. 왜냐면 유전자가 수천 종류가 있는데 근데 이렇게 일단 프리로 해놓고 그 다음에 약간 디퓨전 프로세스를 하면서 그걸 계속 더 파인 튜닝 하는 거라고 생각합니다. 그러니까 이건 그러니까 이건 이제 원 데이터를 가지고 관상을 하는 거고. 근데 디퓨전 그 치료 문제를 계속 풀면서 또 우리가 처음 이걸 100개만 가지고 만들었던 관상 지도가 틀릴 수도 있는 거잖아. 딱 100개, 학생 100명만 가지고 한 거.
약간
우리 프라이어를 주는 거라는 그렇게 하면 되는 거네. 자, 정리해보자. 우리의 목적은 수학 잘 가르치는 건데, 국어 잘 가르치는 건데, 음악 잘 가르치는 건데, 3명을 뽑아야 돼. 새로운 신입 선생님이 오면 딱 관상을 보고 걔는 너는 수학, 너는 과학, 너는 영어 이렇게 정해주는 그걸 우리는 했죠. 근데 현재까지 우리에겐 이렇게 이만큼의 선생님이 있었습니다. 선생님을 가지고 우리가 사실 돈이 많이 없어서 딱 100명만 잡았어. 100명만 가르쳐 봤더니 대충 요 라인은 수학을 잘 가르쳤어야 되고 여기는 과학이고 영어고 뭐 국어고 이렇게 됐어. 대충 요 관사는 수학이고 요까지는 우리가 볼 수도 있는데 사실 자신은 많이 없어. 왜냐하면 100명밖에 안 해봤어. 그거까지가 내 생각에 이거인 거. 100명 가지고 일단 적당히 그래도 이 정도 프라이어. 적당히 하고 근데 디퓨전을 하려면 디퓨전은 근데 데이터 하나만 줘도 데이터 하나랑 그 프리 사이에 몇 스텝이 있어. 1000스텝이 있잖아. 그 1000스텝을 학생을 지도한다고 생각합니다. 그 데이터가 확 늘렸지. 그걸 하면서 다시 이걸 바탕으로 생각해보니까 6는 아니었으니까 2 바꾸고 뭐 이런 거를 할 수 있지. 그러니까 그래서 그렇게 투 스텝으로 가고 있다는 거예요. 약간 이게 프라이어라고 생각하고 디퓨전 모델을 하는데 여기서 러닝 처음에 된 걸 약간 파인 튜닝 한다라고 생각하면 되는 것 같습니다.
그럼 거기서 로스 펑션 데이터가 분류가 키 저희가 관상치도
그거랑 약간 상관없는 거. 아예
V랑 갑자기 예측을 하게 되면 로스가 감소한다면 G의 W를 해서 V가 나와야 됩니다.
근데 그러면 아예 그 키가 아예 거의 없는 상황.
그래서 여기가 약간 프라이어 느낌이고 여기가 포스트리어 느낌이고 네. 어렵다. 그건 근데 뭐 이건 컨퍼런스 페이서니까 그건 안 적었겠지. 그래도 그래도 뭐 하여튼 Aqsa, why don't you go ahead? It's your last. Taking the performance.
Okay, just one. This is just
이것도 한 번 더 뭘 하는구나. Yes. Game is not over.
so basically this is the step in which they know how which input is affecting how much the output. So basically they look here. Epsilon A, we already have seen this, and we have the conditions. So basically, this one is like if we have, as we already discussed, epsilon no drug is like there is no drug in this, and this no free is like it's only drug, and this epsilon in this part is if we have both values. So subtracting these will help us in identifying which condition is getting fulfilled. So basically, SE is a state norm. It is kind of preserving the baseline identity, and ST is the drug norm, which is basically helping us identifying the application of the drug effect. So yes, the ST is like here we have this dose that is going into this function. So basically this is just kind of telling us how much this dose is affecting the final output in the noise.
파인 튜닝 같은 거니까 그래서 넘어가시죠.
So then they compared it with the previous baseline, this method with the previous ones. So if we look here, we can see they have used different matrices to compare the results. And we can see that it is performing, the bold letters are the one where it is performing the best, and underlying ones are the one that are performing the second. In the seconds, so we can see that most of the time it is performing best, and that is the model that is most of the time is performing the highest. So it is just for one day when the model have the unseen combinations of the baseline cells and the drug. So, and the final one is when they have the unseen drug data set, and in this too, we also have like the way this model is performing higher than the other previous model. So in the end, it can be concluded that it can outperform previous models on getting the unseen drug, whether they are introduced with a new drug or there is an unseen combination of the drug and baseline. That's all.
Yes, okay, thank you. Any question?